So I would, if anything, add to that just by pointing out that I think it's a complicated question, in part because it's a two-way street. On the one hand, there may be effects that the intrinsic stability of the mRNA might affect protein production, but so too, conversely, will the action of protein production. That is to say, once an mRNA is initiated and bound by the ribosome, that will tend to promote the stability of that mRNA. And so we observed that in our data as well, that those mRNAs that were bound by ribosomes or had a higher loading were, of course, more stable. And so it may be hard to tease apart, because there are two directions of causality, the relationships between stability of the message and eventual protein production.
Absolutely.
Great. So I'm going to come back to you, Dr. Welsh, it's a fairly long question, so hang in there with me. How can codon usage of naturally highly expressed Ecoli proteins be reconciled with the recent Gustavson data suggesting that optimal design should rather use codons read by tRNAs that are highly charged during amino acid starvation? Do naturally highly expressed Ecoli proteins also have a tendency to have codons of tRNAs that are highly charged during amino acid starvation?
No, actually. So the Gustavson data is they're referring to the paper that we referred to in the slide and the data that we talked about here. And we see almost the opposite. It turns out that most of the most frequently used codons are read by tRNAs that tend to have higher sensitivity to amino acid starvation. The starvation prediction is basically dependent on the ratio of the codon usage frequency to the concentration of measured tRNA in the cell. So things that are used at higher frequency relative to the tRNA amount are most sensitive. Okay, so what we're saying when we're using tRNAs that are less sensitive, we're using tRNAs that are essentially in reserve. They're present at higher concentration relative to their use in basal expression, and we think that may be significant for heterologous expression because we're now flooding the cell with a bunch of new stuff to code. We're flooding the translational machinery with an unnatural level of synthesis and a biased synthesis toward whatever our gene is encoding. So it may be best to tap the reserves of tRNA rather than over tap the ones that are already being used to a large extent in the cell. So yeah, generally it's the opposite. The most frequent codons are more sensitive to tRNAs.
Okay, excellent. I've had a few questions come in about mammalian cell expression, and I'm actually putting together two questions here. Do the principles applied to codon optimization differ for mammalian cell expression, and are there differences in codon bias amongst different types of mammalian cells? So, Dr. Plotkin?
Well, so we've, of course, started to express our constructs in mammalian cells, yeast cells, and insect cells, and I don't think we have enough sufficient data at this point to make any real quantitative response to that kind of question. But I do at least know that the rank order of expression levels in mammalian cells is quite different than in Ecoli cells, and so there may very well be different principles applying to the very same set of synonymous proteins when expressed in a mammalian cell line. And moreover, there is evidence that the codon usage of genes expressed uniquely in different human cell lines differ dramatically between those cell lines, and also evidence from Tao Pan's group that the complement of tRNAs present in different human tissues may differ by tissue. And so there certainly is a possibility for not only different rules of, applying in humans than in Ecoli, but the different rules apply in humans depending upon the tissue or cell type you're looking at. And I think that's exceedingly likely, actually. Dr. Vogel?
Yeah, I don't have much to add, but except for re-emphasizing the fact that in multicellular organisms you deal with different tissues. So if you're then asking about something that's encoded in the genome, it gets very tricky, and you would expect like tissue-specific mechanisms of regulation, no matter what it is.
Okay. So I have a couple of questions that I think are directed at your presentation, Dr. Plotkin. Did you see any differences in translation speed or growth rate between your different GFP constructs?
Sure. Well, certainly with respect to growth rate, we did. We measured a really fairly coarse measurement of growth rate, just the optical density after a certain amount of time post-initiation. And indeed, there were systematic differences between the different GFP constructs in their effect on the growth rate of the cell that was expressing them. Those that had higher CI had faster growth rate despite not having higher protein levels. And our interpretation of that is indeed that elongation is proceeding more rapidly when there's higher CAI, and as a result, there are fewer ribosomes sequestered on those messages and a smaller load on the pool of ribosomes in those Ecoli cells, producing a higher growth rate. We've also measured, at least coarsely, the ribosomal occupancy of different messages as a function of their codon adaptation. And indeed, we have verified that that mechanism seems to match those data, namely the messages that have high codon adaptation have fewer bound ribosomes. And so in some sense, elongation is happening more quickly. That doesn't, after all, produce higher expression levels, but it does certainly make the cell happy.
And the other question was if protein solubility was assessed amongst the different variants that you used.
No, we did not assess solubility yet.
Okay. Quick and easy answer to that one. So I have a question for Dr. Vogel. Often mRNA expression levels do not seem to correlate with protein levels. Is there any way that you can predict this?
Well, one way to deal with this has been mentioned in my talk, where we try and predict this based on sequence features. So that is one thing you want to look at. That has been done for one bacterial system, and people have studied this a lot in yeast. For human, as far as I'm aware, our study is the first one. But as I've shown, it works for about 2/3 of the variation in protein expression, so that is very good, and we're currently working on including other features and generalizing this model, so hopefully this will be useful for other systems as well.
Excellent. A question for you, Dr. Welsh. When codon optimizing your gene, is it recommended to keep the GC content between 30 and 70%? And if you can only optimize one of these factors, in other words, GC content, or have it most optimized, which would you choose?
It's a really good question. It's the problem that really led us to do this work. If you look at different schemes, like matching a GC percentage versus codon bias in another dimension, you quickly run into a conflict. How do you balance these? Which is most important? Is there a sweet spot between them? And when you add in all the different flavors of principles people have added in terms of gene optimization, it really gets complicated. In terms of GC percentage, I think the jury is out on whether GC percentage itself, the total content of GC in a gene, is important, or whether it's the percentage of GC islands, say, or versus AT islands, so the distribution of those nucleotides, which may affect RNA structure or stability. And also whether it's the codon bias that results from varying GC bias. So we don't see a trend with GC bias in our set. We don't have a wide range of GC in our genes, but we don't see a correlation with that. We'd like to actually study that. In our later sets, we have designed some sets that go at that directly, where we're varying codon usage not according to the frequency found in the cells, where we're varying it according to more generic factors like GC versus AT and even purine versus pyrimidine, just to give a more generic diversity in seeing what's important. And we still have more analysis to do there. But my guess is that it's not strictly the GC percentage that's important. It's going to be things that correlate with that in the cases where it is important.
Excellent. Next question is, do longer proteins always have low expression even in lower organisms?
We've observed that length bias in Ecoli, yeast, and human.
Okay. Another short answer. Excellent. Anything else to add?
A bias both in the total protein level, but also critically in the ratio of protein to mRNA. Is
that is correct. Okay. And in fact, at least for the human data set, the bias is even stronger for protein levels corrected by the mRNA level. So you have a negative correlation for mRNA, you have a negative correlation for protein, but that still has the mRNA factor in it. But even if you factor that out, or especially when you factor it out, you have that bias.
Okay, great. Are there known cases where codon optimization of an exogenous transgene inadvertently decreased rather than increased expression levels in mammalian cells? Dr. Welsh?
In mammalian cells, I believe there are. I think there are numerous in the case of E. coli, and I believe in mammalian as well. And that has generally been in using these guesses at optimization schemes. I think there's I don't know if it's published, but I've seen studies where people have taken several I think, mammalian genes in this case, compared wild type to optimized genes, and looked at the statistics. How often did the optimization scheme improve expression? How often did it have no effect? And in some cases, there's a significant number that are actually worse when they optimize, and it just shows you how little we understand what's important. So that's the kind of variability we want to get rid of by just asking the cell and using that to point the way rather than these guesses which clearly are missing the mark.
Okay. I'm going to stay with you, Dr. Welsh, for the next question. Can gene sequence adjustments, for example, codon optimization, impact post-translational processing of the protein, in particular glycosylation? And if so, how might one mitigate this type of impact?
So I think that's perhaps related more to the protein folding question. I mean, the question is, whether, and you may suppose that in some cases that a pause in translation may allow processing to either continue or bypass that. And I don't know of any evidence of that directly, but it's certainly in the realm of possibilities.